Hello everyone. Welcome back to Microbiology Made Easy. Today we are going to learn the carbohydrate fermentation test or sugar fermentation test, a simple yet powerful biochemical test that helps us to identify gram-negative bacilli. Observe the picture carefully. The ability of the bacteria to metabolize carbohydrate is evaluated by three major methods in the clinical microbiology laboratory: the sugar fermentation test, TSI test, and OF test. Each test provides different information about carbohydrate utilization and helps in bacterial identification. The session We will begin the session with a brief introduction followed by clinical significance of the test. Then we will discuss the principle behind sugar fermentation test, the method of inoculation, and finally how to interpret the result obtained in the laboratory. Then fermentation pattern with different pH indicators, associated clinical scenario and points to remember. Let us know about the sugar fermentation test. It is used to determine the ability of bacteria to ferment specific carbohydrates producing acid with or without gas. It is particularly useful in the identification and differentiation of clinically important bacteria. Common clinical conditions where it is helpful. First, neonatal meningitis, urinary tract infection, acute diarrheal disease and wound infection. Applied aspect of carbohydrate fermentation test. First, identification of Enterobacteriaceae. Differentiates lactose fermenters from non-lactose fermenters. Example, Escherichia coli ferments lactose with acid and gas production. Salmonella typhi does not ferment lactose. Second, differentiation of enteric pathogens. It helps to distinguish pathogens causing GI infections. For example, Shigella dysentery ferments glucose only. Escherichia coli ferments multiple sugars with gas production. Third, identification of Vibrio species. Sucrose fermentation helps to differentiate species. For example, Vibrio cholerae ferments sucrose. Vibrio parahemolyticus does not ferment sucrose. Identification of gram-positive cocci. Sugar fermentation profiles assist in differentiating streptococci and enterococci. Fifth, confirmation of biochemical identity used along with other biochemical tests like IMBIC, TSI, urease, citrate, etc. for definitive bacterial identification. What is the principle behind the test? Carbohydrate fermentation mainly leads to formation of acid. The basal media contains peptone water plus 1% specific carbohydrate like glucose, lactose or sucrose. pH indicators used are Andrade's indicator or phenol red or bromocresol purple. pH indicators changes the color of the medium. Durham's tube detect the gas production. Andrade's indicator in most of the microbiology laboratories, this indicator is used. Acid formation mainly changes the color of the medium to pinkish red color. No acid formation leads to yellow or colorless media. Uninoculated media appears very slight pink color, pH 7.1 to 7.2. Methods of inoculation. Inoculate each tubes with pure 18 to 24 hours old culture grown in BHI broth or nutrient broth. Incubate at 37 degrees centigrade for 24 hours. Do not report a negative result before 5 days of incubation. For members of the Enterobacteriaceae family, examine the tube for acid and gas production. To make identification easier, laboratories use color-coded cotton plugs. Green color indicates glucose, red indicates lactose, blue indicates sucrose, and violet indicates mannitol. This color coding system minimizes errors and ensures correct interpretation of fermentation reactions. Result interpretation depends on the pH indicator used in the medium. Different indicators produce different color changes when acid is generated during sugar formation. Example, in the first picture, pH indicator is phenol red. Here glucose and sucrose are fermented. Lactose, mannitol and maltose are not fermented. In case of phenol red, due to acid formation, media turns to yellow. In second picture, pH indicator is Andrade's indicator. Due to acid production, the media becomes pinkish red. Here, pH indicator is bromocresol purple. In this picture, acid formation causes the yellow discoloration of the medium. When is the carbohydrate fermentation test required? The carbohydrate fermentation test is usually required when an organism has been isolated and the microbiologist needs to identify it based on its ability to ferment specific sugars. Let us discuss the clinical scenario. A 5-day-old newborn presented with fever, poor feeding, lethargy, seizures for 2 days. The diagnosis is neonatal meningitis. What to do situation in laboratory? Gram stain shows gram-negative bacilli, LF colony on the McConkey plate and A by A with huge amount of gas production on TSI test. Possible organisms: Escherichia coli, Klebsiella pneumoniae, Enterobacter cloacae. Diagnostic problems: all three organisms may show pink colonies on the McConkey agar, A by A with gas on TSI test. Therefore, TSI alone cannot differentiate these organisms. How carbohydrate fermentation test helps? E. coli ferments glucose and lactose. Then Klebsiella pneumoniae ferments glucose, lactose, sucrose and mannitol. Enterobacter cloacae mainly ferments glucose, lactose and sucrose. Conclusion is the carbohydrate fermentation test provides additional sugar utilization patterns that help to identify the exact species when TSI results are similar. This is especially useful in neonatal meningitis because accurate identification guides appropriate antimicrobial therapy and epidemiological investigation. How is it helpful compared with OF test? A burn patient develops wound infection, culture yields a gram-negative bacillus, TSI K by K, that is no sugar fermentation. Now possibilities include Pseudomonas aeruginosa, Alkaligens faecalis, other non-fermenters. Limitation is the OF test does not tell which sugars are fermented by Enterobacteriaceae. Easy rules to remember. TSI tells does the organism ferment glucose, lactose or sucrose and produce H2S. OF tells mainly does the organism oxidize or ferment glucose. Carbohydrate fermentation test tells exactly which sugars are utilized by the organism for species identification. Thus, the carbohydrate fermentation test is most useful after TSI screening when further differentiation among similar fermenting organism is required. Points to remember: first, place the Durham's tube inverted in the carbohydrate media. Second, gas production is routinely assessed in the glucose fermentation test. Third, most of the Enterobacteriaceae family members ferment glucose. Therefore, the glucose tube serves as the primary indicator of gas production. If TSI test does not show any gas production, then it can be again tested in carbohydrate fermentation media with Durham's tube. In case of large amount of gas production, the media in Durham's tube is replaced by the gas, giving it an empty appearance. Remember, use a sterile inoculating loop for each carbohydrate fermentation tube to prevent carryover of sugars and false positive result. Limitations: it is not a confirmatory test, hence requires test results of other biochemical tests for complete identification. Diverse types of sugars and indicators are available. Lots of media, each containing a specific sugar, is to be made.
If the result is read too late, oxidative deamination may occur, resulting in alkaline pH and causing false negative result. May require more than 24 hours for a positive reaction. Even minor contamination can result in false positive result. Microbiology is not just about studying microorganisms, it is about understanding their unique characteristics, revealing their identity in the laboratory, and connecting them to human health and disease. Keep learning, keep exploring, and see you in the next video. Thank you. Don't forget to like, share, and subscribe the channel for more updates.